In the light-dependent reaction video, we have looked at how light energy is converted into chemical energy and stored in the ATP and NADPH. In this video, we will look at light-independent reaction where energy from ATP and energy from electron in NADPH is transferred into glucose. And this reaction is also called as Calvin cycle. Light-independent reaction occur in the stroma of chloroplast. This reaction do not need light energy to happen, and it involves carbon fixation process where inorganic carbon, where we have here carbon dioxide, is converted into organic compound, that is sugar. Light-independent reaction, or also known as Calvin cycle, involve three phases: carbon fixation, reduction, and finally the regeneration of RUBP. Now let's look at Calvin cycle. As we go through this cycle, please note that the yellow circle represents the carbon atom and the pink circle represents the phosphate atom. First, we have carbon fixation phase. In carbon fixation phase, 3 molecules of carbon dioxide will attach to 3 molecules of RUBP. This attachment is catalyzed by an enzyme called Rubisco. When the carbon dioxide attach to the RUBP, they will produce a 3 unstable 6-carbon intermediate. These 6-carbon intermediate are unstable. Because they are unstable, they will immediately split into half. When they split into half, they will produce 6 molecules of 3-phosphoglycerate, or also known as PGA for short. After that, PGA undergo phosphorylation, where they receive phosphate from ATP. Because we have 6 molecules of PGA, means that we need 6 molecules of ATP to undergo phosphorylation. 6 ATP will donate their phosphate group to the PGA to produce 6 molecules of 1,3-bisphosphoglycerate. After that, 1,3-bisphosphoglycerate undergo reduction, where they receive electron and also hydrogen ion from the NADPH. Again, because we have 6 molecules of 1,3-bisphosphoglycerate, means that we need 6 molecules of NADPH. NADPH donate their electron and also hydrogen ion, so they will be oxidized. They will be oxidized and become NADP+. After the reduction, 1,3-bisphosphoglycerate release one of their phosphate group, okay, and then converted into 6 molecules of glyceraldehyde 3-phosphate, or G3P for short. As you can see in this diagram, how all of the molecules only have one phosphate. From here they have 2, and then from here they have 1 phosphate. Why? Because they released one of the phosphate group to become G3P. So here we have 6 molecules of G3P. After that, 1 molecule of G3P will leave the cycle. Okay, they will leave the cycle. And the other 5 G3P will continue with the cycle with the last phase, that is regeneration of RUBP. The G3P five, the 5 molecules of G3P will undergo molecule arrangement where they use another 3 ATP for phosphorylation to form back 3 molecules of RUBP. Then the RUBP can continue with another Calvin cycle. We need Calvin cycle to occur twice. Why? Because when the Calvin cycle happens twice, we will have 2 molecules of G3P. One Calvin cycle will release 1 G3P. So if the cycle happens twice, means that we can have 2 G3P that will leave the cycle. When we have 2 G3P, means that we can use these 2 molecules of G3P to produce 6 carbon of glucose. In conclusion, to produce 1 G3P, the Calvin cycle will use 3 carbon dioxide, 9 ATP, and 6 NADPH plus H+. So, can you guess how many carbon dioxide, ATP, and NADPH used by the Calvin cycle to produce 1 molecule of glucose? Can you take a guess? And G3P produced by the Calvin cycle will be used to produce carbohydrate such as glucose, amino acid, and lipid in the plant cell. And then the plant cell will be consumed by the consumer such as animals or human. And that is Calvin cycle or light independent reaction.